how a warm brain is actually redefining intelligence. Real-time action predicting system, but connectomes are not really dense. They are surprisingly sparse. Connectome, human brain's connectome is extremely sparse. The attention is actually scattered. Hi everyone. So today our talk is about how a warm brain is actually redefining intelligence, this is especially will be interesting for people who are building real-time action predicting system as AI and in physical AI, physical dynamic systems. So for them it will be very interesting, and we are a team of two. We are trying to solve this research problem as a startup, and we have different skill sets, and we are combining it to find these unknown answers. My co-founder Ranita Jana, Dr. Ranita Jana, she is a computational astrophysicist. Her PhD and postdoc was mostly about simulating large universe, like large scale structure of a universe with 1 trillion nodes, and understanding the relationship between various galaxies. And I am Shapath Das. I have spent the last decade in terms of understanding images and processing images, and also in audience behavioral design, where I had built recommendation systems. So we'll start the talk with the story of connectome. Our story is starting with connectome. Connectome is the most exhaustive point-to-point connection of the entire nervous system, where we are mapping not only the neurons but also synapses. And by looking at these images, actually you might think that connectome is a very dense network, but connectomes are not really dense. They are surprisingly sparse. And here is two sparse network, a sparse and dense network example. One has 13 connection. It has 10 nodes, but only 13s are like connected. By dense network, what we mean is that every node is connected with all the other nodes. Okay, so human brain actually have 86 billion neurons, and human brain is not at all dense connected network. If it would have been dense, then probably we'll have 7.4 sextillion connections between us, between the neurons, but it only have 100 trillions. If you look at the number, that what is the difference between a trillion and a sextillion, a trillion is actually 10 to the power 12, and a sextillion is 10 to the power 21. So we can say that our connectome, human brain's connectome, is extremely sparse. So, but unlike the transformer systems or anything in the deep neural network, artificial neural network which we are using, these are not really following any biological rule in this manner. So though we don't know, I missed this point, that we humans and scientists actually only could map only 0.00002% of the connectome of humans, but there are other species which is available whose entire connectome is actually mapped. So C. elegans is such one species, and from 1986 we know their entire connectome structure. It's the 40 years of research which getting converted into science in last decade. And in the recent time, actually just last year October, another species entire connectome got also mapped. It's called Drosophila, fruit flies, like those flies which we really try to snap it, but we could not. So their connectome we have now. the C. elegans connectome, and the next thing is that the research community is working really hard to map the entire connectome of a mouse. That will be the next connectome which will be available. So C. elegans is particularly interesting because it's the favorite animal across all the labs. It has already won 3 Nobel Prizes directly, 1 indirectly. And these Nobel Prizes was mostly for physiology and chemistry. And why it is interesting, and also it's a very good reference to build a new neural network, because they are fully annotated. The network is fully annotated. We exactly know the name of the neurons, and these are genetically reproducible, and ablation is possible. What is that? Is that scientists can actually identify which are the neurons they want to kill, terminate, and then they do experiments to see that what is the behavioral changes happening. And these are very transparent organisms, so we can actually see what's getting fired, what they are doing. But these are small round worms, but they have behavioral simplicity, but with rich repertoires. That's a difficult word to say. So what do you mean by that? That they have minimum behaviors, but in those behaviors also they have some complexity. So it's a very good animal to study, and that's why it's being studied heavily. Okay, so now let's go into the connectome itself. Like, what is the component of the connectome? Connectome has only neurons and synapses. And C. elegans connectome, this is the only connectome we will be talking today. So has 3 kind of neurons: sensory neuron, interneuron, and motor neuron. You can actually relate with your artificial neural network terms as input layer, hidden layer, and output layer. And this connectome also have these synapses, chemical and electrical synapses. Chemical synapses are like unidirectional connections, and electrical synapses is like bidirectional connection. And why it is important to understand at a neuron level, at the like synaptic level, because if we understand the neurons very granularly, we can actually model an artificial neuron in a same manner. And that is how we can actually build a very robust neural network with these units. So let's also dig into what really happens with neuron. Neuron have mostly just 2 jobs. They sometimes spike and then they take rest. So neuron don't always spike. So whenever there is some input spike coming into neurons, only if it is crossing the neuron threshold, the spike is actually happening. And after the spike, there is a resting period. It's called refractory response, and that time they slowly get charged up and reset, where they are ready to be spiked again. That means that unlike the artificial neural networks right now we are using, which spikes every time, there is, and which can be also parallelized, biological neural networks are not like that. Okay, so apart from this
can you go back to the previous slide?
Okay.
What is the negative graph?
This one? Yeah, so like this is the the resting state of every neuron. So once it has spiked, the membrane potential get decreased. So it's all about sodium potassium. So when this sodium gates and potassium connections are charges are getting decreased, so they reach to this membrane potential level, which is below the resting potential. The membrane's original resting potential, it is below the resting potential. Once it reaches that, it takes some time. But this is not this is also a dynamic time. So every neuron's resting potential at the time it takes to rest is also different.
What is the order of the time, the refractory response time? What is the order of that time?
What is the order means?
As in, a few milliseconds?
Yeah, it will be like 1 to 2 to 3 milliseconds, not more than that. Because I have to move my hands. If beyond that, I will be paralyzed.
What will happen there, the minimum point, which is the negative point?
What will happen?
Yeah.
No, I could not answer your question. So one thing is that I am not able to see from my left eye, so all your faces are blurred. So you have to be louder because I am not able to read also, like usually how human understands.
What will happen when it will touch the minimum point, which is the negative point?
What is the potential there?
Generally, what will happen?
This is the resting potential, and the membrane potential can have both positive and negative values. So after the activation period is over, then the membrane potential gets decayed. And it comes to the resting potential, and sometimes because the time constants are so small that it hits the resting potential and goes negative to it, and then again because of the kind of time constants they have, they again comes to the resting potential. So it always comes to the resting potential, even if it goes to negative values at some point.
Yeah, it will always. Okay. Apart from these neurons and synapses, another thing which is very interesting and which deep neural network has always overlooked is that these neuronal circuits. So these are the 7 circuits of C. elegans which are proven circuits. 5 of them have empirical reproduction results, 2 of them is still waiting for that, and there are actually possibly 40 such circuits inside C. elegans. And the C. elegans, what it does, that using those circuits they do a few of their very minimal tasks. What they do, they if they feel any touch, they do they crawl back or something, or things like that. One such is that the second thing is tap withdrawal circuit. This is interesting because a group of researchers from MIT and Technical University in Vienna, they choose this circuit to build a neural circuit policy based on the liquid neural network. They were building a new neural network architecture, and they started training autonomous car with it. So the training was that a video stream is coming and it need to predict the rotation angle of a steering wheel. That was the training is all about. The task was lane keeping task, and they had also trained conventional neural network, just CNN, also with it, and now we can see the video. Probably it will start. Yeah, so this is the video from that experiment itself. As you can see, this is a deep neural network, CNN, and so it is very hard to identify which neuron is activated to see what, because of the sheer depth of it. And now imagine if this was a transformer, then it's not even possible to even guess what is happening even at a very short timestamp. So the training was happening here is that by giving this footage, if they could understand what will be the steering angle will be, and they had also built attention map, which will come here shortly. So where they tried to identify at each timestamp where the model is giving attention, and all the brightest spots are those spots where the attention is at the maximum. You can see that the attention is actually scattered. There is no pattern in it. Sometimes it is giving attention to the road, sometimes to the surroundings. So this is what actually happening inside deep neural networks, that deep neural networks is working because and it is working, it's still driving. It's not that it's getting derailed. It is happening because of the depth of it. So because it has a depth of neurons, so it is fixing itself. So when they also insert some noise here, so probably some noise here, so we see the result is catastrophic, that there is no idea of where they are focusing. It is so so catastrophic sense that
scattered all over the place.
Scattered all over the place. Yeah. And then when they replace this with a 19 neuron tap withdrawal circuit, which is taken from the C. elegans tap withdrawal circuit, which C. elegans use to navigate when they are facing some obstacle, you can see that the focus is always on the side of the road. So they are actually able to follow the data, when a human is driving, what human focuses, a similar kind of thing. They are also able to focus only the horizon and the 2 lanes of the road. Even when they are actually inserting noise in it, you still see it is not doing any like catastrophic or scattered attention. It is still focused on those areas it need to be focused. So yeah, and now like Dr. Ranita will take further.
Okay, so let's look at the tap withdrawal circuit that Shapath was telling about. It looks like this. It has 2 sensory neurons, 15 interneuron or command neuron, and 1 motor neuron. You can think of the interneuron and command neuron as the central processing unit of the circuit. And this 1 motor neuron was actually predicting the steering wheel has to be go left or right, or what will be the angle it has to be. So as you can also see that this is not a very dense network. All the nodes are not connected to every other node in the circuit, and there are actually 2 types of connections between the source and the target neurons here. Some are marked with green, some are marked with red. The green ones are actually excitatory connections. Whenever there is that kind of a green line, the target neuron's potential, the membrane potential, gets activated when the source neuron fires. But for the red lines, the inhibitory ones, the target neuron's potential does not get increased even if the sensor neuron fires. So yes, so both of them are necessary for the stability of the circuit. The green connections actually helps the neurons to progress or proceed and amplify the signals throughout the circuit, and the red ones you can think it of like gates that tries to stabilize the circuit. And yeah, so and how these neurons are training with the data, and the topology is coming from the C. elegans tap withdrawal circuit, and how the neurons are getting trained is coming from the theory of liquid neural network. So what is a liquid neural network? Liquid neural network is till date the best biologically plausible and compute efficient model to study time series kind of data. You can think of like any sensor data that is coming like a input stream. This is a time series data. And for this kind of data, this liquid neural network works best. This is a form of, this is an improved version of recurrent neural network or RNNs. So let's deep dive into like how RNNs shaped into the liquid neural networks. Let's try to do that. Yes, so recurrent neural networks are kind of architecture where we have feedback loops. Unlike the feed-forward networks, it has the feedback loops which helps for the information to move from time step 1 to time step 2 to move it through a hidden state. So at each step we have a input variable xt and the input from the previous hidden state, and combining these we have the output variable yt. So this hidden state is actually works like a memory of the network, so that it understands the temporal dependence of the data it is feeding, and that is why it understands the temporal dependency of the time series data. But there is a problem. These vanilla RNN or discrete time step RNN, they update in discrete time steps. So if there is a signal, if there is a sensor that is recording a signal at each 5-second interval, the network will get updated every 5 seconds, and it will predict the value also at the same 5-second interval. But the dynamical physical systems do not behave like this. They do not update in discrete time steps. They are continuously evolving systems. So that is a problem, that physical dynamical systems cannot be described by a discrete time RNN. And also, like most of the sensor data in the real life space does not come in the uniformly sampled data. But that is the core assumption of discrete RNNs. And most of the times we get data in irregularly sampled time space data, And this becomes more chaotic when we have like multivariate time series data. We have say 3 signals and 3 of them have different intervals of collecting data, and this becomes chaotic. So what is the way out? Yeah.
So like you said, like while humans do it in a continuous manner, right? But you also mentioned like there's a neurons which actually have like a downtime of in few milliseconds. Isn't then it's plausible that we actually also work in history which has like milliseconds?
Yeah, all of the neurons will not get activated at the same time. They have different kind of activation period and different kind of decay period. So if you feed a time series data, some of the features are like very fast changing, some of the features are very slow changing. So we need different types of neuron to say those different kind of time features. Yeah, please.
So even if one is switched off, we have 86 billion of them. So
yeah, I mean, I'm guessing it's not just one, right? Let's say some 1% of them are switched off.
Not 1%, maybe even less than that. We don't know it. Like, what are the circuits for human?
But like, at least the impression that I got is like different neurons have different sort of the relaxation time.
Yeah, it is dynamic. Like, it's not static that okay, this neuron only has this. It is dynamic. It depends on what data it is processing.
So it changes based on the data?
Yes, itself. Nothing is fixed. Yeah.
Yeah, can you please go to the previous one?
Yeah.
The one before that.
Okay.
So in this, in the previous slide, you had mentioned an analogy of sensory neurons being like the input layer, and interneurons being the hidden, and the motor being the output. So what is the command neurons in that same analogy?
Inter and command neuron, you can think it of it the same way. So there are actually recurrent loops in it. So not much difference, like inter and command are interchangeable, yeah. Not much difference.
Another question.
Yeah.
When you're saying that different, like the sampling becomes a problem and becomes chaotic, what exactly is the problem? Because from what I understand from like first principles, can't you just go to the most granular sample data and then upsample from there?
Yes, if you are if you are doing an experiment in your lab, you can do it. But when different sensor data is coming from different systems and you don't have really control over it. So suppose I am monitoring a patient in ICU. So blood pressure is getting monitored at 1 hour interval, and my blood sugar is monitored in half an hour interval. So that is the protocol or something. So then you have to resample the data, because you want to understand, like you want at a fixed time step, you want all of the data, your blood pressure, your blood sugar, everything, so that you can understand like what is happening. So what they do, if they have like irregularly sampled data, they just throw out the data that do not have those common points, and only take those which have the common points, and they carry forward. But then we actually we are distorting the temporal dependencies, and we are losing a lot of data in that way. Yeah. Okay, so yes, so we understood that we need a continuous time recurrent neural network to model our physically dynamical systems. So 2 Japanese researchers in 1992 came up with this equation. So the physical dynamical systems are best described by differential equations, and this is the differential equation that defines how the state of the neuron changes over time. Here each of the neuron is a dynamical system, and if we have a tap withdrawal circuit with 19 neurons, we will have 19 such equations. So we have to solve this 19 coupled differential equation simultaneously. So what is there? This H is the hidden state variable, tau is the time constant I was talking about, W are the general weights, and I is the input sensory data which we are getting. So for a motor neuron or a hidden state neuron that doesn't have any sensory input, this I will goes to 0. Yeah, otherwise the equation is same. So this equation looks pretty complicated. So we will use this form of the equation for the rest of the talk, where we are saying that this part of the equation, we are replacing it with a nonlinear function f, which has dependency on the hidden state of the neuron, the input data it is seeing, time and theta. Okay. So let's move forward. So this differential equation is actually inspired from neuroscience, how the neurons gets activated and how they decay with time. This is a very famous model for neurons by the famous neuroscientist Louis Lepic, and the model was first published in 1907. This is called leaky integrate and fire model for neurons. And this defines how the neuron's state gets deactivated after it gets fired, how the potential gets decayed with time. Here tau is the time constant, and S you can think it of as a source term that is coming from the input through the synapses. So if you just focus on this part of the equation, it may look very familiar to you, because this kind of equations we have used for radioactive decays or the CR circuit, how the current decays, same equation here. And the solution looks like this, e to the power minus t by tau, and the time constant here defines how fast or slow the neuron gets decayed. So if tau, the time constant, is very large, then the neuron decays very slowly, and these are called slow neurons. And if the time constant is very large, then the neuron decays very fast, and actually they are called slow neurons. And actually, as I was saying, that there are slow kind of features and fast moving features. We need both of these neurons to be there in a handful of numbers in our network to understand the different time features in our data. Okay, so this was the inspiration behind continuous time recurrent neural network. And what Ramin Hassani and Professor Daniele Roos from MIT, what they did, they coupled this equation with another equation which defines how the synaptic model looks. Now this source term also, we have a functional form here, and this comes from a 1952 paper. They coupled these two equations in a single equation where the hidden state with time changes like this. And this is the equation for liquid time constant network. So what is changing from CT-RNN to this CT-RNN to our liquid neural network model? This time constant here, this part is becoming liquid, becoming variable with time and with input data. So this is the effective time constant data, time constant, and as you can see, it is a function of hidden states, input data, time, and theta. So the effective time constant is not fixed for all the neurons. It gets changed as the models see new kind of data or it is experiencing a new kind of environment. So this is different. Yeah. Okay. So now let's see how we can train such a model, such an architecture. The same thing, we also do feed forward and back propagation, but we do it through a differential equation. So this is called adjoint sensitivity method, and this is taken from Chain et al.'s paper from NeurIPS 2018, the neural ODE paper. So we are solving a neural ordinary differential equation in the feed forward manner using any differential equation solver. At the same time, we are also coming up with another equation which is called adjoint state equation, and this equation we are solving from the back of the time. So one solution is moving forward and one solution is moving backward. So at each step you don't have to store everything. Like for differential equations, we are solving the equations in delta t time step, and delta t can goes to very small values. So that the memory does not get explode, we are using this solution. It's a two differential equation solver. One is moving forward and one is moving backward. Okay, so why we say that LNN can better understand the causal structure of the data? Because of these differential equations. Physical dynamics are best described through a set of differential equations. It can predict future evolution of a dynamical system, and it can describe its behavior. Even there is a kind of perturbation or intervention in the system, it can also predict what will happen, and it enhances our understanding of the underlying physical phenomena. That's why it dissects the system's causal structure. Okay, so let's do some more maths. Differential equations actually can form causal structures. We can prove it through some theorems. If we have such a differential equation, say dx dt equal to gx, where gx is a nonlinear function, then and we have an initial value here, then Picard-Lentilov theorem says that this differential equation has a unique solution if this nonlinear term gx is Lipschitz continuous or a very well behaved function. And that leads to a simple Euler method gives us this solution where if we know the value of x at time t, we can get the value of x at time t plus delta t if we know gx, the nonlinear function in this equation. So if we know the differential equation, how the system is getting changed, then we can actually predict the next state of the system through that differential equation. And this representation under uniqueness condition, if this is the only unique solution of this differential equation, forms a causal structure. Okay, so there is another set of models called dynamic causal models, which are mostly used in neuroscience models. And similarly, here the nonlinear function here is f. The same equation that we were using for liquid neural network is the same kind of equation. And if we do a bilinear approximation here, then these three coefficients a, b, c actually tells us how the system changes if certain things change. So what A says us, A says that if the input variable is getting changed, how this function will get changed. Basically, how the differential equation will get changed and how the state will get changed. And B tells us how there is a coupling between the sensitivity between the internal and external interventions, and C tells us how the F is regulating the external input. So that is why the causal structure is coming into place here. And liquid networks are dynamic causal models. As I was saying, that this equation can actually be turned into a bilinear approximation there, and then we can calculate this B and C component, which tells us if there is a perturbation in the system, how the system will react in different conditions. If there is an internal intervention inside the network or there are external intervention through the sensory inputs, how the system will work. We can precisely tell from these equations. Okay, so that is one thing. And I think that is why, because Shapath was saying that for LNNs, the model was only looking at the horizon and the frontal part of the two sides of the lane. For other models, it was not, because it was lacking that causal structure of the data that it was receiving. That is the reason.
What does that mean by causal structure?
Causal structure means if something changes, why it changes. Like if we can say it.
Correlation versus causation.
Yes.
Like we say there's only one solution, so there has to be causation.
Yes. Yeah. Not only causal structure, LNNs also help us mitigate transient perturbations. What happens in CNNs, convolutional neural networks, they process image one frame at a time. They do not have the time dependence or how the input image is getting changed as a time dependent variable. They do not have that information. That is why if somehow one of the image gets corrupted by, say, sunlight or something, then the model gets confused and they cannot understand there is a curvature in the road, because the input image is corrupted here. But what happens in LNNs is that they have this input, image input, this is corrupted, but they have the memory of how it looked like one step back through that hidden state. So it can understand that this is a perturbation, and we don't have to look at it, but we knew that there was a turn in the last step, so it should be there also. So it understands clearly that there is a road curvature, and that is the best thing for time dependency and that understanding the temporal relationship, even if it is an image.
There are also like RNNs, like which basically do that.
Yeah, yes. Okay, so another thing is robustness or generalization. The example Shapath was talking about, the autonomous car driving scenario, there the model was trained in springtime data and it was working equally well for winter time data when the sides of the road were completely covered with snow. That is a very unusual situation because sometimes the model get confused because the models who do not understand where to look at and where not, where to give more attention, these models gets confused. But for liquid neural networks, it was working equally well for these two situations. They did another experiment with a target tracking task for an autonomous drone. They trained the model to follow a red bag in a forest situation environment, and then they were using the same model for different kind of scenario. They checked the model's accuracy in another forest which was not the training one, and then they moved to urban lawn and urban patio, and these NCP and CFCs, they are using liquid neural network in various forms. So comparing LSTMs and GRU, these models, the liquid neural network performs better in all of those scenarios. So in terms of like generalization to out of distribution data and robustness, it was also working very well. Okay, yeah.
In the benchmark, why is it that one model is performing better in one task than CP and CIC?
Which model are you talking about?
Like in the urban norms, CIC got 90% and like it performed better in those benchmarks but not in the previous ones. In the training woods.
Yeah, these are different models, and oh, you are saying why 85% and why 100% here?
Yeah, I mean in the first one, your first model that performed better in training modes and not as good in other models.
Yeah.
The second one performed better in the second scenario. Why is that?
For both of the cases, the training accuracy is more, no?
As compared to between both of them, like one is performing in urban one, it's getting 90%.
90%.
Yeah, so CFC is doing 90% and?
Yeah, and it's not performing as well in the prior benchmark, training modes benchmark.
Like you are saying that CFC is doing 90%, but why NCP is doing 57%?
No, I understood. Like why urban lawn is 90% and CFC is 85%.
Okay.
Yes. So in CFC is what they are doing. It's a approximation of the liquid neural network. So after all, liquid neural networks are also differential equations, and differential equations take time to solve and a bit of compute also. So what they came up with the analytical approximation, a closed form solution of the same equation. So I think that's why it is coming. Like it's not actually the exact solution; it's an approximation of the actual solution.
So I can tell you another thing is that it also depends on which data it is doing. So in a forest like situations, the noise is too much. In an urban situation where they had shot, there was like literally no noise. Everything is very clear in terms of how the model is perceiving. So probably that is also like when they are using a closed form solution to overcome the compute efficiency, so probably that is what hampered.
Okay, should we move forward? Okay. To summarize, liquid time constant networks are novel time continuous neural network for efficiently modeling time series data. LTCs are universal approximators. It has been tried and tested well, and even if the system is very complicated, very complex structure, it comes up with a functional form to model the differential equations. And LTCs are also stable, very stable dynamical systems because of the different time constant, different neurons with different time constants, and also they are variable depending on the input data. They can vary their behavior even post-training, where with the effective time constants are getting changed with respect to the input data. And they are good at learning irregularly sampled data. We have already discussed that. And they are effective for modeling continuous time processes. Yes. So you want to take over?
Yeah. So, so this is actually okay. So, oh, sorry. Yeah. So this is the result for different model they had trained, and this our solution is actually for them. It was that liquid time constant network. So you can clearly see that the difference is in terms of when they are, these are all attention maps. The attention is how a causal model, a model which understands causality, can give more attention from the same data itself. They can learn more features from the same data that where to give attention to if the task is all about lane keeping. So and yeah, and then so we think that the next generation of neural network will be probably will come from India for definitely, and can be more energy efficient, and will be incorporating biologically inspired mechanism. It will be biologically plausible, and this technology will involve sparse network, sparse topology, and it need to be we need to explain each neuron's behavior. If we don't know, then there is no explainability in the system, because in next 10 years when AGI will be there and there will be mostly 90% humanoids in the Indiranagar road, so we don't want to interact with them unless we know that they will not harm us. So that will come from explainability, and we to make sure that they are energy dependent, so that there is no power cord attached to them when they are walking in the road, we need to have dynamic neuron activation signs unlocked. And that means that just like human brains, which has 86 billion neurons, don't fire all the neurons same time, these machines will not use all the neurons at a same time interval. So that is it, and thank you for listening to us.